The following content is provided under a Creative Commons license. Your support will help MIT OpenCourseware continue to offer high-quality educational resources for free. To make a donation or view additional materials from hundreds of MIT courses, visit MIT OpenCourseware at ocw.mit.edu.
We ended last time, we were talking about endogenous activity as one of the determinants of pattern movement. We know very little about that, except a little bit from invertebrates, except we do know about circadian rhythms, which is a type of a more general regulator of movement. Okay, and I've just summarized here the three major types of concepts that have helped us explain patterning in movement. But then there's a couple of things I've added here. Plasticity in these mechanisms. We know about, we're learning about plasticity in synapses, which will affect the first two here, but endogenous CNS activity, especially when single cells generate it, they have these pacemaker proteins that are engaged in this constant cycle where they're changing membrane potential. There's just about nothing known about plasticity, and yet it would be critical if that were a mechanism for timing, and we know what a problem timing is in neuroscience. So I think that's a big deficiency in the field. And I also point out here, my other plus here is that neuromodulators that can alter the state of the overall state of the brain can alter how these patterns are produced. And now that concept is being used in studies of invertebrates. Again, my example here is Eve Martyr at Brandeis, who studied invertebrates for many years and the patterning of how they control pattern movements. All right. The very last topic, which won't take us very long, is the control of the overall state of the brain. I think the chapter is clear enough. You can read it, but I'll just review some major points. What controls it and how many states are possible. Nobody else tries to deal with how many states are possible, so I try a little bit in this book. So we want to know the anatomy of these changes in overall state of the brain, and we mean drastic changes in state, like going to sleep, or becoming aroused from sleep, or from drowsiness, becoming hyperattentive, becoming dominated by some particular feeling or other. Even if it's something simple like a feeling of hunger, it can really change the state of your brain. And a lot of the states that we know humans go through, and animals probably do too, we don't know a lot about. We know some of the correlates because we can record electrical activity. That's what the electroencephalograph does. But it doesn't allow us to specify anywhere near the number of states that should be possible when we go through the anatomy. So there's two very different types of mechanisms that could control a change in brain state. One of them you don't hear much about, and that's, if you look at the top here, the second one, chemical secretions into the cerebrospinal fluid, which can affect a lot of the brain. The study I usually cite for this is a study that's often forgotten about, but if you have a sleeping rabbit and a wide-awake rabbit, and you have a cannula in the cerebrospinal fluid, you take a little bit of the fluid out of the sleeping rabbit and inject that into the cerebrospinal fluid of the awake rabbit. And of course you do the controls. But anyway, the awake rabbit will go to sleep. not studied very much, but I think the cerebrospinal fluid is a medium of conduction in the brain. It probably affects things other than the overall state, but at least we know that it is possible. What we do know more about is the widely projecting axonal systems. You know, if we deal with very primitive animals like Amphioxus, is just one example. Many invertebrates. In fact, if you look at their dendrites and their axons, it looks like they're all very widely spread, or at least many of them. Brains like ours become more compartmentalized, more specific, more specialized. So, but to achieve integration and changes in state, we have specialized systems with widely branching axons. All right. In this question, I ask you to describe 4 axonal systems that are very widely projecting. What are those 4 that we usually think of? So, cholinergic is the first. And we mentioned that when we were talking about brainstem. Remember, I showed those very widely branching axons of the hindbrain. Those are almost always cholinergic. What's a word that tells you monoaminergic? And what are the monoamines? Those are the 2 catecholamines, okay? Dopamine and norepinephrine. Those are both monoamines, but their catecholamines are a subset of monoamines. And what's the other monoamine? Serotonin, 5-hydroxytryptamine. All right, so, and then in more recent years, mainly due to discoveries at Harvard by Cliff Saper, we know about other really widely branching systems that when I was a student, we knew nothing about. All right. So this is that example we had earlier of a widely branching catecholamine using neuron of the reticular formation of the hindbrain. You can see how widespread its distribution is. There are a number of them like this. They're obviously, when they become active, they're affecting the state of the brainstem. And if you're affecting the thalamus here, these midline nuclei they're projecting to have axons that project to the specific nuclei that go to various cortical regions. So they're affecting the state of most of the central nervous system. This is a cartoon of the catecholamine systems. The neuron in the brainstem is like this one, but there's a group of neurons at the base of the brain, nucleus basalis of minor, and there are neurons outside of nucleus basalis that project like this too. They don't include the catecholamine the acetylcholine-containing axons in the striatum. Those are interneurons that use acetylcholine. So all acetylcholine cells are not like this, but these are very widely projected. We usually hear about nucleus basalis in discussions of Alzheimer's disease because they tend to degenerate. Okay, and they project to the entire membrane throughout the limbic system, throughout the neocortex. Here you see them going to the hippocampus. All right, so this is from a medical school book where you see them in the human brain. That's a little bit of optic chiasm that sort of fell off during the histology, so it should be up here. But this is where nucleus basalis is. This is anterior commissure, so you're in front of the thalamus. You're in that basal forebrain region, which we'll be talking more about in the second half of the class. This is all corpus striatum, and here, where you find those acetylcholine interneurons. But the ones in the basal nucleus here, you can see they just indicate they're very widespread projections to the human hemisphere, and that's true of many of the other mammals as well. All right, so here are the monoamine systems: serotonin, norepinephrine, and dopamine, the catecholamines. The first two, serotonin and norepinephrine, we know play major roles in the control of sleep and waking. And more generally, in arousal states, the serotonin axons, which are very widely projecting, begin in these midline nuclei, the nuclei of the raphe in the brainstem, midbrain and hindbrain. You already find those kinds of axons in amphioxus. So they're throughout all of the chordate kingdom. And we know they're important in humans, at least in mood stabilization. We give reuptake inhibitors like Prozac to affect human mood control and prevent the wide swings in mood. They also occasionally cause people to commit suicide, but if I'm giving an announcement on TV, I got to say that, right? I got to say this can cause death, this can cause this, that. I would never take any of those when I hear those commercials. But okay, and then the catecholamines like norepinephrine, we know it's also very important in arousal states, and we know it does play specific roles in switching from one state of sleep to another. And dopamine has more specific effects and has somewhat more limited distribution, not as limited as we used to think, but we know it's very important in reward and reinforcement types of learning. So this is a typical medical school book, medical school illustration of these widely projecting axons. It basically shows you in sort of a cartoon form where the cells are. These are right on the midline in the brainstem. So this is that huge pons in human. You can see these are mostly in the hindbrain. And it shows them, I think this is a later one from Leonard Hamer's books. You can see it projects down to the spinal cord, projects to the cerebellum, projects to the thalamus, it projects everywhere. Very widely projecting, and that's really all you can get out of these. But there is a little more specific knowledge about the serotonin axons. If you look at the raphe nuclei here in the caudal midbrain, you'll see that there's a dorsal and a more ventral raphe nucleus, and they give rise to somewhat different axons. They look different. This is called a thin varicose axon system. And here's a very different kind of axon system. The basket, they call it basket axons. They've got thicker branches, different, larger endings. And if you look at a place like hippocampus or striatum, you see those two structures get only one of those two types. See, the basket axons are going into the dentate gyres. The striatum is getting the thin varicose axons, but they're both going into the neocortex. And we don't know nearly enough about separate functions of those two types of axon for the serotonin system. Okay, and now here's a picture of the norepinephrine. These are amazing cells that you find in this area here called the locus ceruleus. It's named because of the blue pigment there. In rats, there's literally just a few hundred cells, and it projects everywhere. So and they're very thin axons. And the cells aren't all that big, and yet they project more widely than any type of cell in the brain. There is a group in the lateral tegmentum of the hindbrain that also projects, but less widely. It's these locus ceruleus cells that project to the entire forebrain. And it's interesting because if you give a certain toxin, a toxin for the norepinephrine axons, you can, just by giving this early in life, you can destroy all these axons going into the end brain. And when you do that, you don't kill the cells. Instead, they compensate by increasing their projection to the brainstem. So the whole brainstem then gets a super dense norepinephrine innervation. It's a kind of pruning effect of the sort that we've seen in the visual system. All right. And this shows the noradrenaline system in the rat. And the one I put in the book is this one. It's exactly that figure, except I took out a lot for the simple reason that some of these are just mistakes. They're drawn anatomically incorrect. I have the disadvantage of being a neuroanatomist, and I notice these things when I read these summary figures that people mess up a bit. So, but what I am showing here is that even in the brainstem, even though the norepinephrine may be a little less, in spinal cord, cerebellum, and midbrain, it's still there. It's densest in the end brain. All right, and then here's the dopamine system. Now, here's an earlier figure, and here's a later figure. This earlier figure is what you see in most medical books. It shows that in the neocortex, it's prefrontal, and then it also goes into limbic structures. So you see it going into hippocampus, septum, amygdala, and they show it originating in these two structures we've talked about in the midbrain. We've mainly so far talked about the ventral tegmental area when we defined the limbic midbrain areas as the areas closely connected to the forebrain limbic system structures. But then more recent, as these techniques have become more sensitive, they discovered, well, there is a lighter projection that goes all the way back, including into the hippocampus, including into the visual areas of cortex, that does use dopamine. And there's even some going into the midbrain. There's been a little debate about that. I show it coming here from, this is my figure, which was a modification of a summary figure by people that had studied this system earlier and had this kind of pattern. It could be these other groups here give rise to the midbrain dopamine, and we still don't know a lot about the functions of that in all these structures. Notice that there are dopamine cells in the olfactory bulb as well. All right. Well, it turns out those four systems are not the only systems, as I mentioned. We already knew that in the thalamus, the older parts of the thalamus, there are cell groups with very widespread projections. There's one located right on the midline that was studied here at MIT in Nada's lab by Miles Herkenham, who's worked for many years at NIH. He found that this little cell group projects to the entire neocortex, but we're just way up in layer 1. So it's only getting to the dendrites of the neurons all over the neocortex. That's this one. And but since then, and I mentioned Cliff Saper at Harvard, he discovered a number of cell groups that also project very widely. Not quite as widely, perhaps, as the norepinephrine and serotonin. One of them uses melanin concentrating hormone. Another uses the peptides hypocretin and orexin. They were well known because defects in the gene for those substances that are used as neurotransmitters in the system leads to forms of narcolepsy. And some of those connections are still not fully understood. There's another system that uses corticotropin-releasing hormone, a hormone that's used as found, for example, a lot in the amygdala. and it was initially known just from anorexia states, but now we know it is more widespread. This is one picture from those studies. In this particular picture, they show on the right, in blue, the distribution of norepinephrine. Remember, I used blue for norepinephrine in that rat picture before. And here you see it distributed all over the neocortex, all over the limbic cortex down here, and in the hippocampus, throughout the thalamus and the hypothalamus. But in red, on the left, they show these orexin hypocretin cells. And their axons are distributed very widely. They're most concentrated in the hypothalamus and parts of the amygdala. But then, in a more scattered way, you find them throughout most of the rest of the hemisphere. Okay, so it's another widely branching system. So that leads me then to that final topic, well, how many brain states are possible? So what I did for this is just make some simplifying assumptions. I say, let's just take 4 of these systems of the brainstem, and let's say that they just have 3 possible states. No activity, low activity, high activity. And then we add to that the four of these diencephalic state-changing systems and make the same assumption. They have 3 possible states. So that gives you 8 systems. Now, if we assume they can operate independently, and there are actually some interconnections between them, but if we assume they can operate largely independently, then the number of possible states would be 3 to the 8th, 6,561 states. And if you just assume they could have 4 possible states, it goes up to 65,536 states. So that's what I mean when we say that, and these are very easy assumptions to make, because studies of the activity of these neurons indicate that they might have many more possible states than the few I'm assuming here. Just remember, these states are largely independent of sensory, motor, cognitive activity. They certainly interact with emotional activities, motivational states, but they appear to operate pretty independent of those as well. All right. This is just a little about the evolution where Amphioxus is missing. Norepinephrine, he has dopamine and serotonin. Hagfish and lamprey, still very primitive vertebrates, have all of those systems. They may not have all of the others that we mentioned. All right. We've already said what we need to say about those things. So I'll go on to what you guys are most interested in here. This is what I did. Now, first of all, you had that homework 3 where I gave you the worksheets and I told you to do this stuff. Any of that's fair game for the exam. If you did it, you're already prepared there. And then I've selected questions on the book chapters. So for chapters 1 and 2, I only took 7. Okay, chapters 3 and 4, just again a small group of questions. In fact, that goes beyond 3 and 4. That's 5 to 7. So 3 and 4, there's again, it's only 5 questions. So I'm reducing the number of questions a lot. Okay. And I've divided it according to the book chapters. These aren't classes because they don't always match completely. Okay, so I use book chapters here to get these questions. And then, so it still comes out quite a few questions. There's 74 questions. And then I listed some names. Are there any names here you cannot remember? Remember who Fritz and Hitzig were? might be a more difficult one. Those are the guys that mapped the motor cortex for the first time. They did it in dogs and humans during the Franco-Prussian War. They found this one area with the lowest thresholds, the smallest currents they could still elicit movement. And they defined that as the motor cortex. I left Betts out, the guy that discovered the giant cells there. You should certainly remember who Ramon y Cajal was, and that he used the Golgi stain. And was a very comprehensive neuroanatomist. He studied the entire central nervous system and some of the peripheral nervous system as well. His brother, Pedro Ramon, was more of a comparative anatomist, whereas Ramon y Cajal studied mammals. Sherrington, what did Sherrington do? He was a physiologist. Okay. He worked out the properties of synapses before we could see synapses with the electron microscope. He knew about inhibitory and excitatory synapses. He knew about thresholds. He understood how neurons work. In fact, his article on the spinal cord, he studied mainly spinal animals, spinal cats, largely. That is, an animal with the spinal cord transected, and he artificially respirated them for a lot of his studies. He wrote the article in Encyclopedia Britannica on the spinal cord, and I believe a modified version of it is still used in the Encyclopedia Britannica. That's how good it was, even though this guy he was working at the previous turn of the century, early 1900s. Okay, and Otto Lowy, discoverer of chemical transmission, studied the heart, remember? His famous experiments on the accelerator and decelerator nerves of the heart. Hans Spemann, discoverer, or he's credited with the discovery of induction of the nervous system by a specific tissue that turns out to be the notochord. And now we even know the molecule. You remember the molecule? Sonic Hedgehog. Okay. I don't think I put that here. I don't think I did. I did. But Rex had maybe the most difficult one. That was the guy who gave the layers to the spinal cord. And we changed the way we talk about the anatomy of the spinal cord. We use his numbers all the time. Ross Harrison first used tissue culture. So he was one of the first guys to see living growth cones. We first saw those in tissue culture. Carl Lashley is actually better known as the pioneer in physiological psychology, or brain and behavior studies that are experimental in nature. There were, of course, many clinical neurologists before Lashley, but he took the methods of experimental psychology, controlled experiments, and applied it to studies of brain and behavior. Okay, and he was the one who wrote about, I mentioned him twice, actually 3 times in the class, but if you want to find it, most of you can probably search that text, right, and find his name. But the last time he was mentioned was because he wrote that famous paper, The Problem of Serial Order and Behavior, proving that rapid learned movements of humans had to be centrally programmed. So when we overlearn something, we basically are programming our brain that can produce a movement without needing any feedback, anything. He also studied the optic tract projections using a stain for degenerating myelin. Remember what that was? Nauta used silver stains, much more sensitive. It was the Markey method. Anyway, Lashley used that and did a pretty accurate job. He didn't discover the projection of the hypothalamus and other very tiny projections of the optic tract, but he knew the main ones just from that experimental study. You remember who Nauta was? Discoverer of the silver stains used for staining degeneration. And then two more, Levi-Montalcini and Hans Kuipers. Levi-Montalcini discovered what? She did it with Victor Hamburger. They discovered nerve growth factor, the first of a family of molecules we call the neurotrophins, the most well-known growth factors in the nervous system. There are other families as well that are not neurotrophins that have been discovered since, but those are still, when you just say nerve growth factor, you always mean that first one that Levi-Montalcini and Victor Hamburger described. If you just remember her name, that's fine. She died just recently, at age over 100. Okay, and then Hans Kuipers, who with his student Lawrence are well known for their studies of descending pathways, both their anatomy and their function in primates, in monkeys. Okay, now this is the last thing I did. These are just definitions that you should go down to this list and see if you remember all these different words. Most of them you probably will know. Okay? You should be able to find all of those. And so that's it. Are there any parts of the class now that you particularly want to go over? I can go over any of these. We've already gone over the answers to all these questions in the class, so you can find them by searching the class notes. Remember, one was the general introduction, a little bit about glia and methods. So I asked major difference between the track tracing methods of Markey and Nauta. Okay. Nauta being more sensitive because it didn't depend on getting myelinated axons. The unmyelinated ones also. And it was much more sensitive, but not as sensitive as some of the more recent methods. What are those more recent methods? Methods that use axonal transport in various ways, where you use the transport mechanism of the axon to inject something that's then transported. It could be a fluorescent molecule. A lot of them used are fluorescent molecules. They're very easy to use. You don't have to use any histochemical technique. You just inject them. They're transported down the axon. Then you just need a good method of cutting the brain, fixing and cutting the brain, and then looking with a good fluorescent, a very sensitive fluorescent microscope, and look for the fluorescence. Do any of you remember what diffusion tensor imaging is? It uses magnetic resonance imaging, and the computer's programmed to pay attention to diffusion of water molecules. That's right. And water molecules, when it comes to these big bundles of axons in the brain, tends to move down the axon, not across. And they use that to follow bundles of axons. And unfortunately, the cognitive psychologists now think they're studying connections in the brain. They think we don't need animal work anymore. We can study connections in the human brain. I hope you realize how silly that is, because it's not even as sensitive as the Markey technique. Where it doesn't get connections at all, but it does give you the position of the big bundles of axons in the human brain, or any brain that you use diffusion tensor imaging to study. And that's important, because now we can specifically relate these major pathways in human to the studies done in monkey and other animals, but especially the primates, because the humans are, of course, one of the primates. So that's what diffusion tensor imaging is. I asked for its advantages and its limitations. Its advantages is you don't have to kill the animal. Big advantage. And you can trace major bundles of axons. Disadvantage is resolution. You cannot get down to the single cell level at all, and you cannot trace axons all the way to connections. But when you see them coming, and you don't even know which direction they're going, major limitation. But you see axons coming in and out of a gyrus in the brain, you can be pretty sure they're either starting there or ending there, because they wouldn't go up into the gyrus and then go out again. It wouldn't make a lot of sense for them to evolve that way. All right. Some of these are just definitions, like primary sensory neurons and motor neurons, intermediate network neurons. So you remember the midbrain structure that's greatly enlarged in predatory teleost fish? They're depending a lot on vision for their hunting, and they don't depend a lot on learning. So it's the optic tectum, unlearned, visually triggered fixed action patterns. Use the tectum more than any other structure. So they orient towards prey animals, and those neurons are specifically triggered by the types of movement that prey animals make. The frog, another animal. He's a predator, collects insects and worms. He's got a tectum similarly programmed to respond to worms and bugs. If it's a bug moving around like that, the tectal neurons are buzzing away, but the frog just sits there, fixates them. But if that bug stops and he's within range, then the frog attacks. The flick of his tongue and grabs the insect. All controlled from the tectum. What was the major cause of the first three major expansions of the forebrain in evolution? That's number one. It started as an olfactory structure. You see it growing out there at the very front of the brain. And the early cortex was all olfactory. The most primitive animals, the olfactory bulb connects to everything in the forebrain. Okay. Then, what was a later reason that it expanded more? Sorry? Other senses invaded the forebrain. How did they do that? Mainly projections from places like the midbrain tectum, which was very large already. Okay? Any large structure projects all over the place, and it projected right into the tween brain, and tween brain connects to the end brain structures. Okay? Both striatum and cortical. And that, by invading the striatum, it took advantage of the kinds of learning that the striatum had already developed for olfactory guided behavior. That's the speculation. It is very likely to be true. We know that habits in mammals still depend on those pathways through the striatum, except that the inputs to the striatum in advanced mammals comes more from the cortex. It doesn't come from the thalamus, but still, even we have those connections from the older parts of the thalamus right into the striatum. All right, and then the third expansion. Can't hear you. Exactly. With mammals. The neocortex evolved out of the dorsal cortex, which was initially, if you look at the dorsal cortex in reptiles, for example, it's a parahippocampal area. And the parahippocampal areas do, in those animals, get some other sensory inputs. But the big change was new layers, new types of migration into the hemisphere. That led to neocortex, in which, as we know, had a lot of advantages for function and expanded a lot. So those were the three major expansions. It didn't lead to the gigantic nature of the human brain. Well, it did eventually, but there were later expansions that occurred as well. There was a late expansion of the association areas, for example, that led to things like language and other things that we do with our cortex. All right. Remember the name Cynodont? You remember. The mammal-like reptiles, right? And I used early Cynodonts because the late Cynodonts are much more like mammalian brains. So I wanted something that was more like an amphibian brain. And that was the early Cynodonts were like that. But that evolution of those animals what led to mammals. So what expanded when neocortex expands? Animals with really big neocortex, they have other big things too. Cerebellum is one. What projects to the neocortex? The thalamus. So the thalamus gets big too. If cerebellum gets big, what else gets big? The pons gets input from the neocortex and projects to the cerebellar cortex. Okay. So you could say any of those things. Cerebellum, pons, thalamus, they all get bigger as neocortex gets bigger. And if you said something like cerebral peduncle, midbrain, that would actually be true too. There are the axons coming from the neocortex going down to the brain stem, the hindbrain, and the spinal cord. So you know why the face is not part of the dermatome maps? Because the dermatomes are areas of body surface innervated by what? Yes? And by what going into the spinal cord? Spinal nerves, dorsal root of the spinal nerve. Okay. So for every pair of dorsal root, you have a dermatome, one on either side actually. But it doesn't include the face because that is innervated by trigeminal nerve, fifth cranial nerve. Exactly. And remember that peculiarity when we were talking about dorsal column nuclei. I showed a section through the dorsal column nuclei and I said the whole body surface is represented there. Nucleus gracilis, the lowest part of the body. Nucleus cuneatus, the upper part of the body, but not the face. So how's the face represented there? Descending nucleus of the trigeminal nerve. The brainstem trigeminal complex, secondary sensory cells of the trigeminal system, has a descending nucleus that goes all the way into the upper part of the spinal cord. And that's where the face is represented. And that's, if we have pain in the face, it's that part in the upper part of the spinal cord that's being activated. So what's the oldest ascending somatosensory pathway? Spinal reticular. In medical school, you'll learn it's the spinothalamic, because they ignore it. Not all medical school, but some of them talk about spinal reticular also. Remember the raccoon in the Quadi Mundi? There's a figure there. What is the raccoon good at? Manual dexterity. He can get in your garbage cans. He can, the Quadi Mundi can't do that. So what is different about the somatosensory cortex? The representation of the digits of his forelimbs, much bigger in the raccoon. So big that there's a separate gyrus for every digit. The quadrimundi's just got a much smaller little area there. Because the surface area of cortex corresponds basically to dexterity. The same is true in the motor system, for motor acuity. You can talk about motor acuity as well as sensory acuity. I asked you actually to know those 4 basic cellular events that Walpert talks about, because they're all important in many developmental processes. Remember what they were? Adhesion, contraction, growth, movement. They interact, of course. And you can apply them to the growth cone and explain how axons grow. Except you also have to know about membrane addition. Remember the 2 types of cell division, symmetric and asymmetric. In symmetric, they both stay stem cells. In asymmetric, one of them migrates away. The other one keeps dividing. Lateral horn, we talk about dorsal horn, ventral horn, the spinal cord. There's that little bump on the side. Where's the little bump? Thoracic and upper lumbar. Thoracicolumbar or thoracolumbar system is the sympathetic nervous system. They're the preganglionic motor neurons of the sympathetic nervous system. Why are they called motor neurons? Because the axons leave the cord. Well, where's the ganglionic motor neuron? It comes from the sympathetic ganglia, which are all along the side of the cord. All the way from the neck, all the way down to the small of your back. Okay. Plus a few ganglia that sit out in front of the cord, the prevertebral, like the celiac ganglia. All more commonly or popularly known as the solar plexus, because it's a popular place for a boxer to hit another guy. The solar plexus, so he doubles over and loses control of his guts. All right. Sorry, but I've got to use some graphic language here to get you to remember. But okay. If you have trouble and cannot find answers to some of these things, use the forum. Because if you don't, I won't answer. Because I want everybody to get the same information. But you should be able to find answers to all of these either in your notes for the class or right in the book. So I hope that's helpful to you, help you getting ready for this. Now you don't have all this uncertainty. I don't know what's going to be on this exam. You do know what's going to be on the exam. This stuff. I won't tell you how it's going to be written. I might change, for example, just with these definitions or some of these answers, I could put matching questions. But if you know these, you'll certainly be able to answer those. Okay? All right. That's the fairest way I know to get you guys ready. And I wish you all well. We're not going to meet on Friday because I know you are in a hurry to get out of here. And but I will want you to read the chapters on the chemical senses, taste and olfaction. I think it's pretty straightforward in the book. I will answer any questions you have if you have any about them. So that way we can get started with the visual system when you come back.